Hello students, welcome to EPG Pathshala. I am Rashmi Sinha from Indira Gandhi National Open University. Today we are going to talk on module Physiological Role in Sports Performance from the paper Physiology and Sports Anthropology. In this module, we will be able to understand the different physiological variables that influence sports or also the athletic performance, various physiological phenomena and concepts related to sports performance. Also, some of the physiological limiting factors in sports performance. Exercise physiology is a scientific discipline that focuses on how an organism responds to exercise. Exercise represents one of the greatest stresses that an organism can encounter. Therefore, exercise represents an outstanding model for studying human and animal physiology. Most people are familiar with the study of exercise physiology as it relates to sports performance. However, it has become apparent that the study of exercise physiology is also relevant in clinical settings. This realization has emerged from our understanding of how exercise can be used in both the treatment and prevention of multiple diseases including cardiovascular diseases, pulmonary diseases, diabetes mellitus and several types of cancers. Further, the study of exercise physiology helps in understanding the physique forms of a person according to the daily activities or sports activities. We can proceed for various physiological variables separately, either relatively with the amount of body composition and the corresponding sports performance. Physiological variables that influence sports, that is athletic performance. Several of these variables can distinguish between highly trained and untrained subjects. However, within the trained population, there is a considerable variability for each. For example, highly trained athletes tend to have higher VO2 max values than untrained individuals. These high VO2 max values are partly responsible for the better endurance performance of the highly trained runner. There are many examples in literature of outstanding athletes with modest VO2 max values or relatively untrained individuals with outstanding VO2 max values, which could likely be due to the genetic endowment. Percent body fat. Success at the highest levels of many sports requires a specific physique. Body composition is one of the variables related to physique. A very basic assessment of body composition allows for the division of body components into fat tissues and lean tissue. Percent body fat, also depicted by percent BF, is the proportion of one's body weight that consists of fat. There are currently several methods for assessing percent body fat. Most methods for assessing percent body fat are based on relationship between body density and percent body fat. The gold standard, that's the best method for assessing body density, is densitometry, which has traditionally made use of underwater or hydrostatic weighing. Measurement of skin fold thickness also allows for estimation of body density and percent body fat. Measurement of percent body fat determined by skin folds usually correlate well with percent body fat determined by hydrostatic weighing. It should be noted, however, that there are several underlying assumptions made when using skin fold measurements to determine percent body fat. Average percent body fat values for 20 to 29-year-old subjects in the general population are 15.9% for men and 22.1% for women. The percent body fat among athletes of most but not all sports are typically lower than those recorded in the general population. Oxygen consumption, VO2 max. Oxygen consumption is the amount of oxygen taken up and utilized by the body. The oxygen taken into the body at the level of lungs is ultimately used for the production of ATP in the mitochondria of our cells. Because most of the energy in the body is produced aerobically, VO2 can be used to determine how much energy a subject is expending. VO2 can be reported in absolute terms or relative to body mass. Oxygen consumption is dependent on the ability of the heart to pump out blood, the ability of the tissues to extract oxygen from the blood, the ability to ventilate, and the ability to alveoli to extract oxygen from the air. So VO2 is equal to heart rate, that is heart rate in beats per minute, into SV, that is stroke volume, which is the amount of blood pumped out of the heart rate per beat, into A. A VO2 difference is the amount of oxygen extracted from the blood by the tissues. VO2 is VE, that is the amount of air moved in and out of the lungs per minute, into 0.2093 minus FeO2. And what is this? This is the amount of oxygen extracted from the air by the lungs. Resting absolute values tend to be around 0.2 to 0.5 liters per minute in men and 0.15 to 0.4 liters per minute in women. The approximate resting relative VO2 for all individuals is 3.5 ml per kg per minute. Oxygen consumption is most frequently determined using open circuit spirometry. Open circuit spirometry can be used not only for the determination of oxygen consumption but also for the determination of metabolic rate. Maximal oxygen consumption, that is VO2 max, also referred to as aerobic capacity. Maximal oxygen consumption is the highest VO2 value recorded during the maximal exercise. A number of objective criteria can be used post-test to determine whether or not the peak VO2 value during the test can truly be considered a maximal value. VO2 max is thought to be the best indicator of aerobic capacity and therefore the aerobic fitness. It is also a relatively good predictor of endurance performance. However, it is not the only predictor of the performance. VO2 max tends to be higher in men than in women. College-age males have average VO2 max of 45 milliliter per kg minute, and college-age females have a VO2 max about 35. The highest absolute VO2 max values recorded have been in large endurance athletes such as elite heavyweight rowers, whereas the highest relative VO2 max values are typically recorded in small endurance athletes such as cross-country skiers, cyclists, and distance and middle distance runners. VO2 max tests can also be used clinically to assess the type and severity of cardiovascular or pulmonary limitations to exercise. VO2 max can increase with training. An untrained individual may be able to increase VO2 max by such as 15 to 20%. However, in well-trained athletes, increase in VO2 max may not be as great. Fortunately for these athletes, with continued training, they can become more efficient such that they can go faster for given oxygen consumption. Additionally, the percent of VO2 max that the athlete can sustain for prolonged periods of time is also very trainable. Carbon dioxide production. Carbon dioxide is a byproduct of cellular metabolic process. Most of the carbon dioxide given off by the body comes from this cellular respiration. However, during high-intensity exercise, some of the carbon dioxide that the subject is blowing off comes from buffering of the blood. The VCO2 is the response to this exercise. Respiratory exchange ratio, also called as RER, is the ratio of carbon dioxide production to oxygen consumption. At rest and during low-intensity exercise, the respiratory exchange ratio reflects the type of fuel substrates being used by the cell for the production of ATP. For example, an RER closer to 0.70 suggests that primarily fats are being used for the production of energy, whereas RER closer to 1 suggests that primarily carbohydrates are being used. During high intensity exercise, some of the carbon dioxide that the subject is blowing off comes from buffering of the blood and thus no longer reflects solely cellular metabolic events. The normal range of RER at rest and during low intensity exercise is zero point 0.7 to 1, but value may exceed 1.2 during high intensity exercise. Pulmonary ventilation, also expressed as VE. Pulmonary ventilation is the amount of air moved in and out of the lungs per minute. It is dependent on the depth of each breath and the number of breaths taken per minute. At rest, most individuals have pulmonary ventilation of 6 to 10 liters per minute, and maximal exercise values, that is VE max, are in the range of 100 to 170 for most individuals. In elite rowers, values of up to 250 liters per minute have been recorded. VE increases linearly with VO2 and workload until about 60% of maximum. Beyond this point, it increases at a higher rate. Heart rate, HR. Heart rate is the number of times per minute the blood beats. Cardiac output, the amount of blood pumped out of the heart per minute, is dependent on heart rate and stroke volume. Resting heart rate for most individuals is between 60 to 75 beats per minute. Resting heart rates tend to be lower in individuals who exercise on regular basis. Maximal heart rate values are frequently estimated based on subject's age according to the formula HR max is equal to 220 minus age. It should be noted that this formula provides only a very rough estimate of maximum heart rate. Fraction of expired air, that is oxygen, also expressed as FeO2. Fraction of expired oxygen is the percent of expired air, that is oxygen. The air we breathe is 20.93% oxygen, and we typically extract 3 to 6% of air, that is oxygen. Thus, the air that we exhale is usually 15 to 18% of oxygen. Low values of FeO2 suggest that the subject is extracting oxygen well and thus suggests that gas exchange in the alveoli is good. FeCO2 is the fraction of expired air, that is carbon dioxide. The air that we breathe in has very little carbon dioxide, that is 0.03%. As carbon dioxide is produced as a result of cellular metabolism, thus most of the carbon dioxide exhaled comes from metabolic processes. Typically, the air that we exhale is 2.5 to 6% of carbon dioxide. Ventilatory equivalent ratio for oxygen and carbon dioxide. The ventilatory equivalent ratio for oxygen is equal to the pulmonary ventilation divided by overconsumption. At the anaerobic threshold, when a significant amount of energy is coming from anaerobic metabolism, there is an increase in lactic acid in the blood. In order to keep the blood from becoming too acidic, ventilation increases and helps us blow off oxygen carbon dioxide. At this point, VE increases at a higher rate than oxygen consumption, and thus this ratio begins to increase. The ventilatory equivalent ratio for oxygen and carbon dioxide can also be used as an index of ventilatory efficiency. If the subject lungs are very efficient at a gas exchange, the subject will not need a very high VE for a given VO2. The ventilatory equivalent ratio for carbon dioxide is calculated by dividing VE with VCO2. Blood lactate or lactic acid and anaerobic capacity. Lactic acid is one of the products of anaerobic carbohydrate metabolism in the cells. Because it is relatively strong acid, it is usually found in the body in the form of lactate. It dissociates from the hydrogen ions. The amount of lactic acid in blood at the end of a maximal exercise test reflects 1. The intensity of the exercise for the subject, that is, did the subject give maximal effort? 2. the degree to which subject needed to supplement aerobic energy production with anaerobic energy production, and 3. the subject's lactic acid tolerance. It should be noted that the amount of lactic acid in blood at any point in time is dependent on both how much is entering the circulation, that is produced, and how much is being removed from the circulation, that is cleared. For example, endurance trained subjects will tend to have lower blood lactate values at any given running speed or intensity than untrained subjects. This difference owing partly to better ability to use aerobic metabolism in the trained subjects and to a better ability to remove lactate from the circulation. One reason why blood lactate is an important variable to assess is that the hydrogen ions that result from lactate production in the cell are known to cause muscle fatigue via several different mechanisms. An athlete who can perform at high intensities with minimal lactate accumulation in the circulation will be better able to avoid fatigue. On the other hand, during high intensity exercise, the ability to use anaerobic energy system is very important. Thus, it is not uncommon to find very high maximal blood lactate values in athletes competing in high intensity events. Resting blood lactate is typically close to 1 mm. The anaerobic threshold is said to be around 4 mm. One criteria for valid VO2 max test is value exceeding 8 mm, and elite athletes competing in events lasting 2 to 6 minutes have been reported to have values exceeding 25 mm. To perform the exercise is coming from aerobic energy systems. At intensities above this threshold, anaerobic energy production is needed to supplement aerobic energy production. For an average person, the anaerobic threshold occurs at 50 to 60 of VO2 max. In highly trained runners, the anaerobic threshold is typically between 75 and 90% of VO2 max. There are several ways of assessing anaerobic threshold. Individuals with high VO2 max typically also have high AT, that is the anaerobic threshold, but the relationship between AT and VO2 max is highly variable. Having a high AT is probably more important for long distance runner, and having a high VO2 max is probably more important for mid distance runner. To be successful, mid-distance runners also must have a great ability to tolerate high concentration of lactic acid. If it appears that VO2 max is as high as it can go for a particular athlete, then one way the athlete can continue to improve performance would be to modify their training to focus on improving their AT. The classic way of assessing the AT is by assessing blood lactate continuously throughout the test and identifying the point where blood lactate begins to accumulate. However, it is not always convenient to take blood lactates throughout the test. Fortunately, there is non-invasive means of estimating the anaerobic threshold using the ventilatory response to exercise. The ventilatory threshold can be determined using several methods. One of these methods is to identify the point where VE and VO2 max begins to continuously increase. Running economy and mechanical efficiency. Running economy reflects how efficient an athlete is. An athlete who is more efficient can run faster for a given oxygen consumption. It also means that if two athletes are running at the same VO2, the one who is more economical will be able to run faster. Running economy improves over time and is typically greatest during the peak transmission season. Physical activity and health. These changes in muscle and in cardiorespiratory function are responsible for increases in both VO2 max and lactate threshold. The endurance trained person can thus perform at considerably higher rates of work than the untrained person. Increase in VO2 max generally range from 15 to 20% following a 6 month training period. However, individual variations in this response are considerable. In one study of 60 to 71-year-old men and women whose endurance trained for 9 to 12 months, the improvement in VO2 max varied from 0 to 43%. The mean increase was 24%. This variation in response may be due to part to genetic factors and to initial level of fitness. To illustrate the changes that can be expected with endurance training, a hypothetical sedentary man's pertaining values have been compared with his values after a 6-month period of endurance training and with the values of a typical elite endurance runner. Responses to endurance training are similar for men and women. At all ages, women and men show similar gains at strength from resistance training. Maintaining fitness and muscular strength. Muscle strength and cardiorespiratory capacity are dependent on separate aspects of exercise. After a person has obtained gains in VO2 max by performing cardiorespiratory exercises 6 times per week, 2 to 4 times per week is optimal frequency of training to maintain those gains. Further, a substantial part of the gain can be retained when the duration of each session is reduced by as much as 2/3, but only if the intensity during these abbreviated sessions is maintained at 70% of VO2 max. If training intensity is reduced by as little as 1/3, however, a substantial reduction in VO2 max can be expected over the next 15 weeks. In previously untrained persons, gains in muscular strength can be sustained by as little as a single session per week of resistance training, but only if the intensity is not reduced. Environmental conditions. The basic physiologic response to an episode of exercise vary considerably with changes in environmental conditions. As environmental temperature and humidity increase, the body is challenged to maintain its core temperature. Generally, as the body's core temperature increases during exercise, blood vessels in the skin begin to dilate, diverting more blood to the body's surface where body heat can be passed on to the environment. Evaporation of water from the skin surface significantly aids in heat loss. However, as humidity increases, evaporation becomes limited. These methods for cooling can compromise cardiovascular function during exercise. Increasing blood flow to the skin creates competition with the active muscle for a large percentage of cardiac output. When a person is exercising for prolonged periods in heat, stroke volume will generally decline over time, consequent to dehydration and increased blood flow in the skin. Heart rate increases substantially to try to maintain cardiac output to compensate for the reduced stroke volume. High air temperature is not the only factor that stresses the body's ability to cool itself in the heat. High humidity, low air velocity, and the radiant heat from the sun and reflected surfaces also contribute to the total effect. For example, exercising under the conditions of 32 degrees Celsius air temperature, 20% relative humidity, 3 kilometers per hour air velocity and cloud cover is much more comfortable and less stressful to the body than the same exercise under conditions of 24 degrees Celsius air temperature, 90% relative humidity, no air movement, and direct exposure to the sun. Hydration and endurance exercise. Sweating is normal physiological response to prolonged exercise required for dissipation of heat produced during energy metabolism. Unfortunately, this natural occurring response can also result substantial fluid loss and impaired endurance performance. Inadequate fluid balance throughout prolonged bout of exercise or training sessions results in several deleterious physiological events including increased heart rates and temperature. Research has suggested that rising body core temperatures may cause fatigue in muscles and central nervous system. Dehydration also results in higher heart rate values for the same submaximal intensity due to decreased stroke volumes resulting from the lower blood plasma volumes. It is possible for the heart rate values to approach maximal levels genetic limitations to endurance exercise. The physiological limitations to endurance exercise examined thus far, including VO2 max, economy, lactate threshold, substrate availability, utilization, and hydration, may all be influenced by another factor, genetics. Whereas researchers have reported a genetically regulated upper limit to individual VO2 max values. These findings indicate that regardless of training, volume, or intensity, 10 to 30% of the variability in VO2 max is genetically determined. The genetic influence on VO2 max has been attributed to both central and peripheral factors, with genetic effects on cardiac output having been reported to be as high as 50%. Similarly, training improvements in economy and lactate threshold are parameters that are also genetically regulated. Genetic differences in muscle fiber proportion are also commonly found in individuals. Slow twitch muscle fibers, which is, we just talked about the genetic difference in muscle fiber proportion, it is also the slow twitch and fast twitch. So slow twitch muscle fibers characterized by more mitochondrial mass and enzyme levels than fast twitch muscle fibers have increased capacity for mitochondrial respiration. Elite endurance athletes generally possess high percentages of slow twitch muscle fibers in muscle, contributing to their respective endurance exercise. In fact, elite marathoners have been reported to have greater than 90% slow twitch muscle fibers in muscles of legs. The advantage of more slow twitch muscle fibers includes greater mitochondrial capacity, increased oxygen consumption, and increased performance in endurance exercise. High correlations between slow twitch muscle fibers and endurance performance have been reported in both running and cycling. Individual fiber type proportions also genetically regulate the training adaptability to the physiological parameters of VO2 max, economy, and lactate threshold. Some ideas about human performance. Most models of sports performance focus on distance running and endurance cycling. First, there are excellent records and standard events. Second, there is a comprehensive physiological data on a large number of elite athletes. Third, it is possible using treadmills and cycle ergometers to reasonably stimulate in a laboratory what is happening during actual competition. We should also note that for the purpose of this review, we assume that environmental conditions are ideal and do not add any additional challenges to physiological regulation, most notably the challenges associated with high altitude or high environmental temperatures. Several well-accepted concepts have emerged related to endurance exercise performance velocity, and the first component issue is the level of aerobic metabolism that can be maintained during race, that is the performance. The upper limit for this is maximal oxygen uptake. This is usually achieved during relatively large muscle mass exercise and represents the integrative ability of the heart to generate high cardiac output, total body hemoglobin, high muscle blood flow, and muscle oxygen extraction, and in some cases the ability of the lungs to oxygenate the blood. By the 1930s, very high values for VO2 max in athletes were observed and identified as a marker of elite performance. Champion endurance athletes have VO2 max values of between 70 to 85 ml kg-1 minute -1, with values in women typically averaging about 10% lower due to lower hemoglobin concentration and higher levels of body fat. VO2 max values 50 to 100% greater than those seen in normally active healthy young subjects are seen in champion endurance athletes, and the most striking adaptation to the training that contribute to this high VO2 max values include increased cardiac stroke volume, increased blood volume, increased capillary density, and mitochondrial density in trained muscles. Of these, the most dominant factor is the high stroke volume. Once it became reasonably clear that elite runners had high value for VO2 max, it also became clear that for events lasting beyond 10 or 15 minutes, most of all, the competition was performed at an average pace that did not evoke VO2 max with much of the 42-kilometer marathon run approximately 75 to 85% VO2 max, while 10 kilometers is performed at 90 to 100% VO2 max and 5 kilometers at a close to VO2 max. Along these lines, it has recently been shown that maximal aerobic metabolism can decline acutely during the course of 5 to 8 minutes laboratory performance bout. This decline is caused by a fall in stroke volume and accelerated muscle fatigue due to reduced blood and oxygen delivery and increased anaerobic metabolism. This does not invalidate the concept of VO2 max but rather indicates that the maximal rate of aerobic ATP resynthesis during the race is dynamic and that truly accurate models of energy turnover during actual competition would require instantaneous measurement and calculation of fluxes through multiple metabolic pathways. Lactate threshold. Based on the concepts above, the question then became what fraction of VO2 max might be sustained for the periods of time extending to several hours, that is the marathon, and what is the rate of glycolysis in active muscles at this rate of mitochondrial oxidation. This question led to observation showing a curvilinear relationship between blood lactate values during exercise and disease of effort, and led to the concept of the rate of aerobic metabolism maintained during performance bout. Can be better described by degree of muscle glycolytic stress reflected in lactate population in addition to VO2 max. Running speed or power output on a cycle ergometer increases in untrained subjects. There is typically no sustained rise in blood lactate concentration until about 60% of VO2 max is reached. There is a long history of investigation about what causes this rise in blood lactate levels and how lactate does or does not contribute to fatigue. The important points include: 1. The initial appearance of blood lactate is not synonymous with hypoxia in the skeletal muscle, and 2. The lactate molecule per se does not cause muscle fatigue. Individual record of treadmill velocity and VO2 versus blood lactate concentration in subjects capable of breaking 2:30 hours for a marathon. What appears to be occurring is that maximum rate of fat oxidation is inadequate to meet the ATP demands of muscle contracting at moderate and high intensities. This causes intracellular signaling events to occur which stimulate glycogenolysis and glycolysis, and ultimately the rate of pyruvate delivery to the mitochondria progressively exceeds the ability of the mitochondria to oxidize pyruvate, and this leads to accelerated generation of lactic acid. The associated hydrogen ion is then likely culprit in muscle fatigue and also activates group 3 and 4 skeletal muscle afferents that evoke important cardiovascular and autonomic reflexes. While the physiological determinants of the lactate threshold are extremely complex, they are determined mainly by the oxidative capacity of the skeletal mass. This capacity is highly plastic and can essentially increase more than twofold in trained skeletal muscle of human or animal who engage in 20 to 120 minutes of training at requisite intensity. This more than doubling of oxidative capacity is one of the factors that is linked to high lactate threshold values seen in elite endurance athletes. As noted, these elite athletes have VO2 max values that are 50 to 100% above those seen in normally active sedentary young people, and their lactate threshold occurs at a higher percentage of their VO2 max. This means that in elite athletes, the absolute oxygen consumption that can be generated for a long period of time before reaching the lactate threshold is essentially doubled, allowing sustained running speeds of 20 km/h or cycling power outputs of 400 watt. Other key factors that reduce muscle fatigability and lactate production during exercise at 85 to 90% VO2 max, when only a fraction of total limb muscle mass is simultaneously recruited, is the quantity of muscle mass that the athlete can recruit to share in sustaining power production. Elite cyclists appear capable of rotating power production through 20 to 25% more muscle mass throughout a 1 hour bout of cycling, thus reducing the relative power production and stress on a given fiber. Additionally, this power sharing among fibers would also reduce the glycolytic stress and lactate production per fiber due to more total mitochondrial sharing for a given rate of aerobic metabolism. These factors should operate in a complementary way that reduces the stress per mitochondria and muscle fiber. An exercise extends beyond about 2 hours, the problem becomes one of the fuelability as the glycogen content in skeletal muscle becomes depleted and the modest ability of active muscle to take up glucose from blood can limit the rate of oxidative ATP generation and thus the pace that can be sustained. In some, not all, subjects, the associated reduction in blood glucose evoke frank symptoms of hypoglycemia that limit the ability of the individual to continue exercising. Other highly trained subjects show remarkable resistance to hypoglycemia, and for their athlete muscle, glycogen depletion is probably more important. In response to these events, a number of pre-competition dietary strategies and during exercise energy replacement regimens and products have been developed. When these are used in optimal manner, muscle glycogen stores can be augmented by 40% before exercise and hypoglycemia can be avoided with the net effect being that the duration of exercises at about the lactate threshold can be extended by about 1/3. Performance VO2 and anaerobic metabolism. Without practical direct calorimetric methods to measure instantaneous rates of heart and work production during endurance exercise, the best practical estimation of the rates of actual metabolic energy production and ATP turnover is obtained from measures of oxygen consumption during an endurance performance bout. During marathon running, the relative amount of anaerobic metabolism is small, yet in the events lasting 13 to 30 minutes, it will be significant, contributing perhaps 10 to 20% of total ATP turnover. This anaerobic contribution to ATP turnover during endurance performance bouts is noted and has classically been estimated from measures of post-exercise consumption. However, the rate at which this energy might be generated and consumed is difficult to estimate in a definitive way. Efficiency. The next factor that makes an important contribution in endurance exercise performance velocity has been termed economy or efficiency. In the above sections, we outlined how VO2 max and the lactate threshold operate to determine performance. VO2. The next question then is how much speed or power can be generated for that level of oxygen consumption. The oxygen cost of endurance running at a given speed can vary 30 to 40% among individuals. When cycling at a given power output, the oxygen cost and thus gross mechanical efficiency also varies from one person to another, but by somewhat lesser amount compared with running. Gross mechanical efficiency when endurance trained cyclists generate 300 watt can vary from 18.5 to 23.5%, and it appears that more than half of this variability is related to percentage of type 1, that is the slow twitch muscle fibers of the vastus lateralis muscles. The efficiency with which the chemical energy of ATP hydrolysis is converted to physical work depends greatly on velocity of sarcomere and muscle fiber shortening. Type 1 fibers display greater mechanical efficiency when cycling at cadences of 60 to 120 rpm. Therefore, it is not surprising that elite endurance cyclists typically possess some higher percentage of type 1 muscle fiber given that they are more efficient. Although type 1 muscle fiber in untrained humans possess a higher mitochondrial density compared with type 2 fibers. Type 2 ones are the fast twitch. It is important to note that with the intense interval training, mitochondrial activity can be increased to equally high levels in both fiber types. Thus, with intense endurance training over years, the main functional advantage of type 1 fiber appears to be efficiency when cycling rather than total oxidative ability, although type 1 fiber seems to retain a greater ability to oxidize fat. It is also of note that many champion cyclists chose pedal cadences of about 90 rpm. This is a cadence that may actually increase whole body oxygen consumption slightly for a given body power output from VO2 minimum, which is usually at 50 to 60. In a comprehensive engineering physiology analysis of this problem, Hansen et al. Noted that subjects with high level of myosin heavy chain self-selected higher pedal rates, and these rates closely match the rate of peak mechanical efficiency. In this context, they speculated that motor control pattern in these subjects might favor a faster cadence so that relatively low total muscle forces per pedal stroke could generate the needed power so that the higher force motor units could be conserved. Running is a more complicated movement than cycling in that it elicits more stretch on the muscle prior to contraction, and there is more potential to capture mechanical energy in the elastic elements of tissue. However, although there has been long-standing interest in identifying the biomechanical and anatomical factors that allow one person compared with another to expend 30 to 40% less energy per kilogram of body to move at a given velocity, the etiology of differences in running economy generally remains a mystery, and biochemical descriptions of running are not good predictors of running economy. Elite endurance runners typically possess a predominance of type 1 muscle fiber, and it would seem logical that they are more mechanically efficient at velocities of distance running. However, running and walking economy has not often been highly associated with the person's percentage of type 1 muscle fiber. This agrees with the idea that running economy reflects the interaction of numerous factors, including muscle morphology, elastic elements, and joint mechanics, in the efficient transfer of APT to running speed. The extent to which cycling efficiency or running economy can be improved with the training has also been of long-standing interest. Until recently, it was generally believed that cycling efficiency and running economy did not improve much with training. At best, running economy might sometimes increase slightly over a course of 2 months when explosive type weight training is added to an endurance training program. However, the conclusion that efficiency does not change with training was based on cross-section comparisons of relatively small number of endurance athletes. Some ideas about physiological limiting factors. 1. Marathon. In 1991, these concepts were used to predict that a much faster world record for the marathon was physiologically possible based on the idea that marathon running speed was essentially predicted by the equation VO2 max into lactate threshold percentage into running economy. When reasonable estimates of the best values were recorded for these three parameters were used in this equation, a predicted optimal marathon time of around 1:45 hours emerged. Even when assumptions about wind resistance were added, times well under 2 hours seemed possible. In retrospect, one overlooked possibility was that the VO2 max values used in the estimates came from laboratory studies typically conducted while the subjects ran up a grade of 5 to 10%, and these values may be approximately 10% higher than those seen during level running. However, even if the highest VO2 max values seen during graded running protocols are not attainable during level running in many people, there are high enough VO2 max and lactate threshold values that might result in sustainable oxygen uptakes, which in combination with outstanding running economy would generate a marked improvement in current world record time. These comments reinforce the conclusion of this earlier modeling effort that either these are unknown factors that operate at a high speeds that make such time not achievable, or that for some reason best-in-class values for every factor are unlikely to occur in the same person. The second factor, genetics. The role of genetics plays in an attainment of world-class status and truly elite athletic performance. There are a number of studies showing that key elements of response to training in sedentary persons is widely variable and has a genetic component. There have also been reports suggesting that common single nucleotide polymorphism might be overrepresented in either groups of elite endurance athletes or in sedentary subjects that respond most to training. The most notable example is the idea that I for insertion variant of angiotensin converting enzyme ACE gene is overrepresented among elite endurance athletes. However, in the largest cohort of elite athletes who have been both rigorously phenotyped and genotyped, this association has not been confirmed, and to date there are no genetic markers identified in humans that have been clearly shown to be more frequent in elite endurance athletes. Another interesting example relates to the gene encoding for the skeletal mass isoform of AMP deaminase. There is a common mutation of this gene that may be associated with lower exercise capacity and trainability in untrained subjects. While the frequency of gene may be lower in elite endurance athletes, there are still a number of elite performers who carry it, so it does not appear to preclude the attainment of elite status, and there is at least one example of an elite performer with essentially no AMP deaminase activity. Finding genetic markers that are strongly predictive of either success in endurance athletic performance or somehow preclude it is likely to be a daunting task because of the many cultural and environmental factors that contribute to success in sport, the many physiological factors that interact as determinant of performance, and the heroic nature of the training required. Ideas about culture are highlighted by the observation that while East Africa runners currently dominate international competition, previously athletes from Australia and New Zealand, preceded by Eastern Europeans, and even earlier, the Finns showed remarkable level of success. This geographical diversity argues against a simple genetically based set of answers to the problem of elite performance in endurance competition. In a parallel way, there is a low signal to noise ratio for many proposed genetic factors that might contribute to multifactorial medical conditions like heart disease, diabetes, hypertension, and clear causal association between genotype and phenotype are slow to emerge. 3. Endurance. Though the physiological mechanisms regulating endurance performance is quite complex, the main factors limiting prolonged exercise have a straightforward interpretation. To continue exercise for extended duration, sustained muscle contraction must be maintained and is dependent on the continuous provision of both oxygen and the fuel. Although each of the physiological limitations is modifiable through endurance training, it is important to recognize that genetic factors play a tremendous role in the trainability of limitations and the capacity. It is therefore critical for coaches, fitness instructors, and personal trainers to recognize all of the physiological components limiting endurance performance as they design exercise programs to improve endurance capacity despite the submaximal nature of endurance exercise during severe dehydration. Fourth, dehydration. Although dehydration is a naturally occurring physiological limitation to endurance exercise, it can be countered to a certain degree by adequate hydration practices both prior to and during endurance exercise. Currently, there are many different approaches and products used by endurance enthusiasts for pre-exercise and exercise hydration, including the ingestion of solutions comprised of water, salt, simple carbohydrates, electrolytes, and glycerol. Let us sum up this module. Physical activity and sports performance has fundamental role in prevention and treatment of chronic disease. The precise measurement of physical activity is key to many surveillance and epidemiological studies investigating trends and associations with disease. Public health initiatives aimed at increasing physical activity rely on the measurements of physical activity to monitor their effectiveness. Physical activity is multidimensional and a complex behavior to measure. Its various domains are often misunderstood. Inappropriate or crude measures of physical activity have serious implications and are likely to lead to a misleading results and underestimate effect size. Methodologies particularly suited for use in epidemiological research are reviewed with particular reference to their validity, primary outcome measure, and consideration when using each in the field. It is acknowledged that the choice of method may be a compromise between accuracy level and feasibility, but the ultimate choice of tool must suit the stated aim of the research. A framework is presented to guide researchers on the selection of most suitable tool for use in specific study. Thank you.